Why has it been so difficult to make progress in treating many of the devastating diseases in our society that our industry has labeled as intractable? Decades of efforts have gone on by attempting to create small molecules and biologic drugs that look at the problem as one to correct a single protein or maybe even a single pathway event. For examples, in diseases such as heart failure and Parkinson's disease, where more than 30 million individuals will be affected worldwide, we have had little impact on this disease at present. At BlueRock, we believe the root cause that is common to these types of diseases is the loss or complete dysregulation of a specific cell type in our body. Once this cell type is lost to the system, there really is no drug that can fully recapitulate all of the functionality of this exquisite machine. But what if we could replace these cells? This is exactly what we're doing at BlueRock. Our focus is on building and replacing this lost cell type. Hello, and thank you for your time today. My name is Seth Ettenberg, and I'm the Chief Scientific Officer at BlueRock Therapeutics. Our mission at BlueRock is to develop authentic cellular medicines to reverse devastating diseases. And we go about this mission based on our proprietary iPSC platform. In the first instance, our technology that we're building, we refer to as authentic cell replacement, in which we seek to replace the lost cell, fully restore its function, and thereby reverse the course of disease. But since these cells are manufactured from an indefinite, controllable supply, and we have access to and the capability to use gene editing technology, we are also engineering these cells at the master cell bank stage to enhance the therapeutic capabilities of these cells and broaden their application in both rare and chronic disorders. We refer to these cells as engineered authentic cells. We think of ourselves as an engineered cell therapy company pioneering a novel class of therapeutic cellular therapies. We refer to our platform as Cell Plus Gene. Our current disease focus is in 3 areas: neurology, immunology, and cardiology. The evolution of our technology, of course, did not happen in a vacuum. In fact, much of the key accomplishments and the lessons we are building from started out decades ago in the first wave of cell therapy, in which donor-derived cells were engineered with viral particles like in CAR T. And of course, this has transformed medicine. In these foundational achievements, both the source and the production with random viral engineering made these products highly variable. But it is from this success of the field that we can envision ways to better control these variables. At BlueRock, we have built an induced pluripotent stem cell platform to better control the source materials going into manufacturing and better assess and characterize and quantify and release the final drug product. This predictable nature and scalable method allows us to use gene editing technology to deliver a more homogeneous, controlled, and ultimately scalable product. Beyond our current efforts, we see a future where gene circuits born from the parallel efforts in synthetic biology can now be built into our products to enhance the cell's natural capability with added therapeutic control. While most of us know of using iPSCs as a basis for a manufacturing platform, let me take a moment to introduce you to our process. We begin with high-quality, well-characterized, healthy donor starting materials, in most instances from the blood, and from this, we reprogram these mature cells into iPSC using our proprietary reprogramming methods. From this point, we can decide to either engineer or not. In some indications where we can combine immunosuppressive drugs and we're replacing cell types that may lie behind immune privilege barriers, we can move directly into expanding these cells and differentiating them into their final mature therapeutic type. But this ability to scale and genetically alter the cell early on in our manufacturing process, for instance, to evade detection by the immune cells in the patient, such as in a universal off-the-shelf off-the-shelf product. Clearly, the ability to cloak or hide cells from immune detection for some period of time and in some diseases for the lifetime of the patient is critical to the success of our field. At BlueRock, we are making great progress to designing and testing such products. Our company was catapulted into the field and our capabilities enriched by our founding scientists that have spent decades perfecting the study and the controlled process of differentiating iPSC. As I mentioned earlier, we're focused on 3 disease areas. Our founders represent clear expertise in these areas and are critical to our success. Our efforts in neurology, which I will go into more detail in a few slides, have been built from the fundamental learnings and the art of Dr. Lorenz Studer's work. While we are making our first entry into the clinic using his methods to produce midbrain dopaminergic neurons, we are pursuing several different cell lineages that are deemed crucial for a healthy brain and central nervous system. With the help of Dr. Bruce Blazar, we are building key regulators of the immune system, which will allow us to pursue settings of acute and chronic inflammation with suppressive cells such as Tregs and macrophages. Of course, the flip side of these cell types can be used in settings where tissue is required to be deleted, such as in cancer, or reshaped, such as in fibrosis. And finally, in the setting of heart failure, we are working closely with Dr. Gordon Keller, who has been one of the primary sources of data and understanding for many cell types, including authentic ventricular cardiomyocytes. But producing the specific cell type in the lab is not enough. And as many of us have come to learn, precisely how you go about this production and maintain the intricate biology of the cell that you are scaling is not yet a linear, predictable path. Therefore, at BlueRock, and with the coordination and continued support of our parent company, Bayer, all along we have been investing in and building across the entire manufacturing chain. From the very start of BlueRock, we have maintained the control of every step of our process, from iPSC derivation through master cell bank production to early clinical materials from our site in Toronto. And coming soon, for the control of the raw materials for our genetically engineered products, we'll be pursuing this in Cambridge, Massachusetts. And most recently, to the commercial application with a BlueRock Bayer Cell Culture Center in Berkeley, California for commercial products. At BlueRock, we have a rich and diverse pipeline across different cell types from our 3 core disease areas. With over 6.5 million Americans suffering from heart failure and with a greater than 50% mortality rate in 5 years from diagnosis, there are very limited treatment options. Here we're advancing our efforts in the differentiation and product production of a left ventricular myocyte. Equally devastating, there are more than 80 million individuals worldwide who suffer from autoimmune diseases, 24 million in the United States alone. Most of these have no cure and may require lifelong treatments. Here we're advancing several cell types, such as macrophages and Tregs. Moreover, there will be more than 60,000 Americans diagnosed with Parkinson's disease this year, and there's no disease-modifying treatment or hopes for a cure. Our recent announcement of an acceptance of an IND in collaboration with Memorial Sloan Kettering, based on the work of Dr. Lorenz Studer, we are poised to demonstrate the ability to replace the cell of origin in this disease, the cell that supplies dopamine, at the right location and with the correct self-regulated control. Let me share a few slides to walk you through this endeavor. Parkinson's disease has high unmet medical need. It's the second most common neurodegenerative disorder that gets diagnosed. It's, of course, a chronic and progressive disease where patients lose both motor and non-motor symptoms caused by the loss of the dopamine-producing neurons. Symptoms can be characterized by tremors, rigidity, cramping, and dyskinesias. And as I mentioned, there are few and not adequate drugs available to these patients. The medications affect more of the patients at the early stage and become less effective as the disease progresses. Treatments such as deep brain stimulation carry the risk of infection, stroke, and seizures, are costly, and can require follow-up surgeries. Of course, there are gene therapy approaches that are very promising, and these approaches are attempting to induce non-native cells to express dopamine. Our collaboration to build the best-in-class differentiation process and cellular products started from the initial NISTEM funding and the elegant work of Dr. Studer's laboratory. This work was shared in 2 recent publications and demonstrates how we are producing both a highly pure specified dopaminergic neuron, eliminating much of the contaminating cell types from the earlier protocols, and demonstrate a native functionality such as spontaneous signaling or bursting of neuronal activity. In these recent papers by the team demonstrating some of the key preclinical data for this work, here I'll share just 2 snippets of that data, which use this differentiation protocol to produce midbrain dopaminergic cells. These cells were used in a model of Parkinson's disease where 6-hydroxydopamine lesioned nude rats were implanted with 400,000 cells per animal. On the left is a representative immunohistochemistry image from these animals 3 weeks post-transplantation. This demonstrates clear evidence of engrafted human cells as stained by H&A or human nuclear antigen. These cells are double positive for FOXA2, a lineage-defining transcription factor, and TH, tyrosine hydroxylase positive cells, the enzyme that's responsible for converting tyrosine to L-dopamine, the rate-limiting step to the production of dopamine. We saw complete reversal of the impairment in these animals, of these hemilesioned animals, which are induced to spin or rotate by chemical stimulation. A clear decrease in all the animals with the human dopaminergic neurons implanted were seen starting at about 5 months post-implantation. This makes sense as our cells are implanted as neuronal progenitors and fully mature in the correct tissue. As we recently announced, we now have an ongoing phase 1 trial. This is intended to be a multi-center trial with a single dose escalation in advanced Parkinson's patients. We will enroll about 10 patients that are still responsive to L-DOPA treatment, but do not maintain or are not getting adequate relief from this treatment. An important element in our strategy to treat patients that are still capable to respond to dopamine. Of course, as this is a phase 1 study, our primary objectives will be safety and tolerability. We will follow the cell's engraftment through PET imaging, and we hope to be able to assess preliminary efficacy by assessing motor and non-motor neuron function, as well as quality of life at both 1 and 2 years post-implant. We see our trial as a significant leap forward from previous attempts using fetal cell sources, and furthermore, we have learned to better understand the patient population by assessing an ongoing Parkinson's disease natural history study at Weill Cornell called the Atlas. So what's next? After producing midbrain dopaminergic producing cells and treating Parkinson's disease, what are the other cell types or indications that are possible across our portfolio? We and others have clearly demonstrated the capability to develop various cell types from the myeloid derivation. Specifically, we see macrophages as the platform within our platform. What I mean by this is that macrophages play a key role in our bodies. They are present in every tissue in our body. We often name them by this presence in this tissue, such as microglia in the brain or Kupffer cells in the liver. This pliable and multifunctional cell can be used and naturally home to the areas of inflammation or tissue injury. These cells can exist in either a suppressive state or an excitatory one, depending somewhat on the environmental cues that they receive. We can again use these cells in their native state for authentic cell replacement or engineering them to lock them in one phenotypic state, say suppressing an exacerbated immune response, or the potential engineering of these cells to produce therapeutic payloads to deliver to the right locations. There will be broad therapeutic potential from one industrialized bioprocess for these cells. While I don't have time to run through specific examples, we have demonstrated and produced cells that carry scFvs, such as anti-tau and Alzheimer's disease models. We've also used these very same cells to produce specific cytokines in oncology applications or immunosuppressive applications in autoimmunity settings, where the site and the extent of delivery is essential to the therapy. Additionally, we have used these cells to replace missing enzymes in such settings such as lysosomal storage disease, where not only is the production of the complicated enzyme difficult, but the delivery of this enzyme to the right location is essential as well. There is still much to do in the areas that we expect many in the field to contribute to and excel in, and I expect BlueRock will continue to collaborate on several of these areas, such as continued optimization and industrialization of our ongoing production of our cells. We are seeking to build what we call smarter cells, which will enhance their functionality through genetic modifications, such as those gene circuits that are coming from synthetic biology. We will seek to expand our gene editing capabilities as well as gene modification strategies. And finally, we expect to collaborate and work and continue to work on novel differentiation and discovery efforts and expand our therapeutic footprint in the coming years. Thank you very much for your attention, and I look forward to any questions you may have.